Okay, let's unpack this. Imagine someone diagnosed with Parkinson's facing that progressive loss of control, suddenly finding real hope.
Hmm.
Hope in a treatment that might actually slow or even reverse the degeneration.
Yeah, or picture someone with severe burns and the wounds heal dramatically faster.
With less scarring, too, right? All thanks to these microscopic biological tools.
Exactly.
So today we're taking a deep dive into, well, one of the most exciting frontiers in medicine, stem cell therapy.
And their even tinier offshoots, exosomes, mighty little things.
Right. Our mission for you today is really to cut through the complex science, get to the core of what these treatments are.
Where they're being used, how they're changing lives.
And also the promise, but the challenges too. The very real challenges they present.
Yeah, absolutely.
Think of it as your shortcut, maybe, to understanding this medical future that's, well, it's unfolding right now.
And we've pulled together quite a stack of recent research, lots published just this year, last year.
Good stuff.
Oh, yeah. Journals like Stem Cell Research and Therapy, Signal Transduction, and Targeted Therapy. Even some great accessible pieces from places like Live Science.
Okay.
And what's really fascinating is how they paint this whole picture. Not just the what, but the why and the how.
Right. The mechanics of it.
Exactly. From the basic biology, the fundamentals, all the way to cutting-edge clinical trials.
And it touches on gene editing, too.
And
ethics.
all of it. Gene editing, the ethical dilemmas, the regulations. We'll be looking at the main types of stem cells, the surprising power of these exosomes.
There are applications all over the body.
Yeah, how tech like CRISPR fits in and those really critical safety and ethical points that, well, everyone's grappling with. Researchers, society, all of us.
Okay, so let's start right at the beginning then.
Yeah.
When we talk about stem cells, it still sounds a bit sci-fi to many people.
Mm-hmm.
What actually are they? What makes them so different from, say, a skin cell or liver cell? What's the source of this incredible potential?
Well, at their core, stem cells are special because of two really powerful interconnected abilities.
Okay.
First, they can self-renew. That means they can just keep making more copies of themselves almost indefinitely.
Like a biological copy machine.
Pretty much. And second, they have this unique capacity to differentiate.
Meaning they can change?
Exactly. Transform into various specialized cell types. Think of them as the body's master cells or maybe raw materials.
Got it.
But they're not all identical. There's a kind of hierarchy, like a developmental family tree.
Okay.
So right at the top, you have totipotent stem cells. These can form a whole organism, everything, including the placenta and support tissues.
Wow, but that's very early on.
Extremely brief, yeah. Right after fertilization. Then you get pluripotent stem cells.
Like embryonic stem cells, ESCs.
That's right. They come from the inner cell mass of a very early embryo. Now, these can differentiate into any cell type in the human body, nerve, muscle, skin, you name it.
But not the whole organism.
Correct. They can't make those crucial support tissues on their own. And then a really huge breakthrough happened with induced pluripotent stem cells, iPSCs.
Yes, iPSCs. These are adult cells, right? Like
Yeah.
skin cells.
Exactly. Adult cells reprogram back to that pluripotent, that master cell state.
And iPSCs were just such a game changer, weren't they? Because they offered a way around some of those very significant ethical issues with embryonic stem cells.
Precisely. A massive leap. It opened up these incredible paths for personalized medicine, but without the ethical weight of, embryo destruction.
Okay, so that's pluripotent. What else?
Moving down the hierarchy, you find multipotent stem cells, often called adult stem cells.
More specialized.
Much more specialized, yes. They can only differentiate into a limited range of cell types, usually related to the tissue they come from.
Like blood stem cells making blood components.
Perfect example. They're basically for maintenance and repair throughout life. And you find them in lots of places: bone marrow, fat tissue, even your dental pulp.
Huh. Didn't know that.
Yeah, but the key thing for you to remember is that all these types share that incredible ability to self-renew and differentiate. That's the core of their regenerative promise.
It's amazing, like these internal repair crews ready to go.
Yeah.
But our sources also talk a lot about something even smaller, exosomes, like tiny messengers sent up by the stem cells. What makes them so powerful? Maybe even more useful than the whole cells sometimes.
You've definitely hit on a really crucial point, and it's an area that's just exploding with research right now.
Okay.
See, while stem cells themselves have immense potential, they do come with some practical and safety
It's
considerations.
like rejection.
Yeah, there's always a potential for an immune reaction, or the challenge of making sure they actually survive and integrate properly where you put them.
Right.
And there's even a very low, but it's a present theoretical risk of, well, uncontrolled growth if any undifferentiated cells accidentally get transplanted.
Oh, okay. So that's where exosomes come in.
Exactly. Stem cell-derived exosomes are emerging as this really viable and often potent alternative.
So what are they exactly, these exosomes?
Think of them as tiny, super sophisticated biological mail packets, or maybe message bottles sent between cells.
Okay.
They're a type of extracellular vesicle, basically lipid-bound spheres, typically just 30 to 150 nanometers across, incredibly small.
Nanometers. Wow.
Yeah, tiny. They actually originate from inside the cell, from something called the endosome, and they carry this really precious cargo: proteins, lipids, nucleic acids like RNA.
And they do the same job as the stem cell.
Crucially, yes, they inherit many of the therapeutic effects of their parent stem cells, but they deliver it in a much safer cell-free package.
Cell-free. So these little packets of information can do the work without all the baggage, let's say, of the whole cell. That sounds like a pretty big advantage.
It really is. Their advantages are, well, numerous.
Such as?
First, they're largely non-immunogenic. They don't really trigger a strong immune response like whole cells might.
Okay, less rejection risk.
Right. And they also lack that infusion toxicity you sometimes see with cell therapies. Practically, they're generally easier to get, easier to store.
And because they aren't living cells,
Mm,
no tumor risk.
exactly. Free from tumorigenic potential. That addresses a major safety concern right there. Plus, a lot of the ethical debates around cells like ESCs just don't apply.
Hmm.
Essentially, exosomes act as the active agents. They deliver the therapeutic signals without the risks you get from transplanting living cells.
And they can travel further.
They're incredibly stable, yeah. And they can even cross biological barriers that whole cells struggle with, like the blood-brain barrier.
Yeah.
Makes them ideal for targeted delivery.
That's fascinating. So these super small, super effective packets. But how on earth do researchers work with something so tiny, almost invisible?
Yeah, good question.
How do you even identify them precisely? Characterize them, store them, make sure they're safe and potent.
Characterization is absolutely critical. You can't use them therapeutically without it. So researchers use a combination of really sophisticated methods. They need to verify the size, the morphology, that's the shape and structure, and crucially, what's inside, the cargo profile.
Okay, how do they measure size?
For size, we use techniques that can literally count and measure nanoparticles suspended in liquid. It's almost like shining a laser through a cloud of dust and analyzing how the light scatters.
Oh, clever.
Yeah, it ensures they're in that therapeutic size range. For morphology, shape, specialized electron microscopes are key.
To actually see
To
that.
visualize them in incredible detail, yeah.
Yeah.
Confirm that spherical shape, check their integrity.
And the cargo, the important stuff inside.
Right. To understand their therapeutic power, the proteins, lipids, genetic material researchers basically do sophisticated molecular detective work.
Like what?
Techniques like Western blotting to pinpoint specific proteins, or mass spectrometry to get this detailed molecular fingerprint of everything inside.
So you know exactly what molecules they're carrying.
Exactly. It confirms their potency. And for storage, cryopreservation is usually the way to go. Ultra-low temperatures, like -80 Celsius.
To keep them stable.
Yeah, it's considered optimal to maintain their structure and content for a long time, though there's also growing interest in lyophilization freeze-drying.
Freeze-drying, like astronaut food.
Sort of. It allows for easier storage, maybe even at room temperature for a bit, especially if you add cryoprotectants. Makes them much more accessible for clinical use, potentially.
It's clear the science of just handling these tiny tools is incredibly precise. So, okay, we've got these amazing cellular and subcellular tools. Where are they actually being applied, or where's the most promise? Our sources mentioned a huge range.
Oh, absolutely. The applications are incredibly diverse. It really spans almost every major medical field, which is why it's so revolutionary.
Let's hit some highlights. Orthopedics.
Yeah, orthopedics is a great example. Exosomes are showing tremendous promise for healing fractures and treating conditions like osteoarthritis.
How do they help with fractures?
They promote osteogenesis, that's new bone formation, and angiogenesis, which is the growth of new blood vessels, both absolutely critical for repair.
And osteoarthritis. That affects so many people.
For osteoarthritis, they seem to promote chondrogenesis, new cartilage formation. Plus, they reduce inflammation in the joint and can even modulate the immune response, so it's like a multi-pronged attack on a really common debilitating condition.
Wow, actually rebuilding and repairing joints and bones? That's huge.
Mm.
What about neurological conditions? Those are often so tough because the brain doesn't regenerate easily.
Right, the neurosurgical field is another area with just immense potential exactly because of those challenges.
Like stroke?
Yeah, for ischemic stroke where blood flow to the brain gets cut off, stem cell-derived exosomes show neuroprotection. They basically shield brain cells from damage.
And they help recovery.
They seem to, yes. They also promote neurogenesis, that's new neuron growth, and studies show improvements in cognitive function, spatial memory.
What about traumatic brain injury, TBI?
For TBI, again, exosomes have been shown to promote functional recovery, reduce neuroinflammation, boost neurogenesis. Sometimes they even outperform the parent stem cells in improving sensorimotor recovery in studies.
Better than the original cells. Interesting.
It can happen, yeah. And think about Parkinson's disease. Research suggests exosomes might attenuate neurotoxicity, protect those vital dopamine-producing neurons, and potentially slow the disease's progression.
Delivering healing factors right to the brain.
Exactly. That targeted delivery is key.
That's genuinely remarkable. Offering hope where, traditional treatments often just manage symptoms.
Yeah.
And autoimmune diseases, where the body attacks itself, like in MS.
For autoimmune conditions, the immunomodulatory properties, the ability to rebalance the immune system of mesenchymal stem cells, or MSCs, are really central.
So they calm the immune system down.
Essentially, yes. Reduce that destructive self-attack. For aggressive forms of multiple sclerosis, MS, hematopoietic stem cell transplantation, HSCT, is actually a primary approach now.
With good results.
Studies show remarkable reductions in relapses, improvements in disability scores. It's quite powerful.
And other approaches for MS.
MSC-derived neural progenitors are also showing promise, especially in progressive MS. Early studies suggest they're safe and can lead to sustained, though sometimes variable, reversal of disability over a couple of years.
And Crohn's disease, too.
Yes, in Crohn's, particularly for fistulizing disease, bone marrow MSCs and adipose-derived MSCs stem cells from fat tissue are being used. They help promote fistula closure and tissue repair.
So again, calming the immune system and promoting healing.
Both at the same time, yeah.
It's just clear these therapies offer this really deep level of cellular repair and modulation. And you mentioned earlier a functional cure for sickle cell disease. How does gene editing fit into all this?
Gene editing.
Yeah.
Particularly CRISPR-Cas9, it's just a massive game changer accelerating this whole field.
How so for sickle cell?
Well, for conditions like sickle cell and beta thalassemia, clinical trials using CRISPR-edited hematopoietic stem cells, patients' own blood stem cells, edited, have already resulted in these long-lasting increases in fetal hemoglobin levels.
Which counteracts the disease.
Exactly. And it's not just managing symptoms. Researchers are genuinely calling it a functional cure because it corrects the underlying genetic defect in the patient's own stem cells. It's profound.
Wow. Beyond direct cures.
Oh, yeah. The technology is used extensively just to model genetic illnesses in the lab much more accurately, to identify new drug targets, develop therapeutics for all sorts of things, Duchenne muscular dystrophy, some cancers.
They're with organoids, too. Mini organs.
Yes. Another incredibly exciting area, organoids are basically these miniature, multicellular, self-organizing structures grown from stem cells in 3D cultures.
Like tiny brains or livers in a dish.
Pretty much. Mini brains, mini livers, mini kidneys. They provide much more physiologically relevant platforms for studying diseases, discovering drugs, screening for toxicity.
Reducing animal testing.
Dramatically reducing the need for animal testing, yes. They're helping us understand diseases on a level we just couldn't before and test potential treatments with incredible specificity.
Okay, the progress is just staggering, from fixing single genes to growing mini organs. But inevitably, such powerful tech raises huge ethical and safety questions.
Absolutely. Society needs to grapple with these.
What are the main concerns that researchers and regulators are focusing on?
It's a crucial part of doing this responsibly, and the sources definitely highlight several critical areas.
Like with embryonic stem cells.
Right. First, with ESCs, there's that long-standing ethical conflict about destroying human embryos to get the cells. Views differ deeply on the moral status of an embryo, of
And
course.
the iPSCs were meant to solve that.
The discovery of iPSCs initially seemed to bypass some of that, since no embryos are destroyed, but
There's always a but.
always. iPSCs brought their own ethical complexities. Concerns about their unlimited differentiation potential, maybe misuse in human cloning scenarios, or that risk of tumor formation if any undifferentiated iPSCs get transplanted.
So safety is still a concern even there.
Yes. And while mesenchymal stem cells, the adult ones, are generally considered quite safe, you still need continuous monitoring. Long-term follow-up is essential to rule out any possibility of adverse events down the line.
So safety isn't just about the immediate aftermath of the treatment. It's long-term. Decades, maybe.
Exactly. We vitally need long-term data. What if implanted cells don't mature correctly, or they age prematurely, or maybe even create misfolded proteins or plaques years later, potentially leading to something like dementia?
Big questions.
Huge. It makes long-term monitoring critical, and it raises other questions, like should recipients of certain stem cell therapies be restricted from donating blood or tissues later?
I had thought of that.
Transparency in research, careful informed consent, these are paramount. Donors need to fully grasp these potential downstream uses and risks.
Like their cells being sequenced. Or used in animal models.
Precisely. Genomic sequencing, putting human cells into animal models for testing. Some donors might object based on religious or deep ethical beliefs. It boils down to ensuring autonomy and respect at every single stage.
And then there's the other side. The unregulated clinics making wild promises. That seems incredibly dangerous.
Unfortunately, yes. That's a really significant concern. One of the toughest ethical challenges is the sheer number of clinics popping up offering unproven, unregulated stem cell treatments.
Preying on vulnerable people.
Often, yes. Exploiting patients with false hope, marketing totally unscientific therapies for serious, even terminal diseases. They might use questionable cell sources. They definitely lack proper authorization from the FDA or EMA.
And the risks are real.
Significant patient risks. Infection, adverse immune reactions, potentially even death. It's not only harmful to patients, it massively erodes public trust in the legitimate science.
Which we need.
Absolutely. It just underscores how urgently we need robust regulatory oversight, clear ethical guidelines, and really informed public engagement to steer the responsible development of these powerful tools, make them accessible but safely.
So quite a journey we've taken through this microscopic world. From the incredible versatility of stem cells, the ethics around where they come from, to the targeted power of their tiny messengers, the exosomes.
Yeah, and seeing how these tools, supercharged by gene editing and organoids, are already making this profound impact.
Repairing bones, healing wounds, fighting neurological diseases, autoimmune conditions.
A huge range.
But we've also had to confront those vital ethical questions and the very real safety challenges that come with science that's groundbreaking.
And if we zoom out, connected to the bigger picture, what really stands out is just the sheer breadth of this, the transformative power.
Orthopedics, neurosurgery, autoimmune diseases, regenerative medicine.
It's truly global potential. The market's projected to hit, $44 billion by 2032. It's massive.
Which brings up that big question you raised.
Right. How do we make sure this revolution is both responsible and equitable?
Because the science is advancing so fast.
Exactly. The scientific leaps need to be matched by clear ethical frameworks, solid regulation, and an informed public conversation. We need to ensure the benefits reach everyone who needs them safely and fairly.
Just imagine that future, though. A diagnosis of Parkinson's or maybe a chronic injury isn't met with resignation anymore, but with genuine hope for a personalized, precise, really effective cellular treatment, maybe even that functional cure like we're starting to see for sickle cell.
It's a powerful vision.
So as you ponder those huge implications, maybe consider this.
Yep.
What role do we as an informed society actually play in guiding how these powerful new medical tools are developed ethically, equitably, and safely?
A question for everyone.
Keep digging, keep learning, keep asking those important questions about where medicine is heading. We really hope this deep dive has given you some powerful insights to chew on.